Hello students, in this lecture let us learn about nucleus, structure and function. Let us now see the introduction. The presence of nucleus in the eukaryotic cells is the principal feature that distinguishes them from the prokaryotic cells. Most eukaryotic cells generally have one nucleus, but some cell types, example the mammalian RBC or red blood cells, lack nuclei, while other cell types, for example the osteoclasts, have many nuclei. Sometimes the nucleus is lobated, as in most granulocyte types of white blood cells, where they can be bilobed, trilobed or multilobed. The nucleus is a membrane-bound organelle that houses the cell's genome and also serves as the main control center of the cell, where DNA replication, transcription and RNA processing takes place. The nucleus is bound by a double membrane called nuclear envelope that encloses the nuclear matrix that contains the nuclear lamina, which is a cytoskeleton-like network that provides mechanical support within the nucleus. The nuclear envelope is impermeable to large molecules, and therefore larger molecules such as protein and RNA are actively transported by carrier proteins present in the envelope that forms nuclear pores. The nucleus also contains many nuclear bodies, for example the nucleolus, which are made up of particular parts of the chromosomes, RNA molecules and specific proteins. The nucleus provides a cellular compartment in eukaryotes that maintain the genetic material and also controls the activities of the cell by regulating gene expression. Let us now see the structure of the nucleus. The nucleus occupies a large percent of the total cell volume in most eukaryotic cells, and it is the largest organelle in animal cells. The nucleus is filled with nucleoplasm, and the fluid component of nucleoplasm is termed the nucleosol, which are similar to the cytoplasm and cytosol respectively. The internal structure of the nucleus includes the nuclear envelope, chromatin, nucleolus and nuclear matrix as the main components, and these structures organizes the genetic material and also localizes specific functions of the nucleus to discrete sites inside the nucleus. The detailed structure of the nuclear components will be discussed in the following sections. First, let us take up the nuclear envelope and pores. The nuclear membrane or the nuclear envelope is made up of an inner and an outer membrane. The two membranes are parallel to one another and separated by 10 to 50 nanometer. The outer membrane is continuous with the membrane of the rough endoplasmic reticulum and contains ribosomes on its surface like the rough endoplasmic reticulum. The space between the two membranes of the nuclear envelope is called the perinuclear space and is continuous with the lumen of the rough endoplasmic reticulum. The nuclear envelope serves as a barrier to diffusing movement of molecules between the nucleoplasm and the cytoplasm. However, the envelope contains nuclear pores or channels composed of multiple proteins called nucleoporins. A cell may have many pores, which is about 3000 to 4000 in mammalian nucleus. The nuclear pores are made up of around 50 proteins in yeast to about several hundred proteins in vertebrates, and the pores have molecular weight of about 125 million Daltons. Each pore contains an 8-fold symmetric ring-shaped structure at the region where the inner and outer membranes of the envelope fuses. A structure called the nuclear basket, which is attached to the ring, extends into the nucleoplasm, and another set of filaments extends into the cytoplasm. Although the average diameter of the pore is about 100 nanometer, the gap available to molecules to freely diffuse is only about 9 nanometer wide, which allows only small water-soluble molecules to pass through it. Larger molecules such as proteins, ribosomal subunits, and some DNAs are transported actively into the nucleus by a family of transport factors known as karyopharins. The karyopharins are of two types: importins that mediate transport of molecules into the nucleus, and exportins that mediate transport of molecules out of the nucleus. Several proteins, including those that play a role in genome structure and function, such as histones, DNA polymerases, RNA polymerases, transcription factors, splicing factors, and many others are targeted for transport into the nucleus by specific amino acid sequences called nuclear localization signals that direct their transport through the nuclear pore complex. The next structure is the nuclear lamina. The nuclear lamina consists of a network of intermediate filaments that provide mechanical support on the internal face of the nuclear envelope in animal cells. The nuclear lamina is composed of lamin proteins which are synthesized in the cytoplasm and transported into the nucleus. A less organized network of intermediate filaments provide support on the cytosolic phase of the nuclear envelope and contains lamins such as imerin and nesprin that binds to the cytoskeleton and provide structural support. Some lamins are also present in the nucleoplasm of the interphase nucleus, where they form a structure called nucleoplasmic veil. The lamin monomer contains an alpha helical domain around which two monomers coil around one another to form a dimer structure called a coiled coil. Two such dimers arrange in an antiparallel manner to form a tetramer called a protofilament, and 8 protofilaments arrange laterally to form a twisted rope-like filament. These filaments are dynamic structures and are assembled or disassembled depending on the competing rates of filament addition and removal. The nuclear lamina provides structural support to the structures present inside the nucleus, and they also serve as sites for anchoring chromosomes and nuclear pores. The next structure are the chromosomes. The DNA or the genetic material of eukaryotic cells are visible inside the nucleus of dividing cells as highly condensed structures known as chromosomes. While in other phases of the cell cycle, the linear DNA molecules are organized as thin threads of DNA-protein complexes known as chromatin. In addition to the DNA present inside the nucleus, eukaryotic cells may contain DNA in two other organelles, that is the chloroplast and mitochondria. The next structure is the nucleolus. The nucleolus is the largest nuclear body found inside the nucleus. It is a densely stained and membraneless structure that forms around tandem repeats of DNA coding for ribosomal RNA or rRNA or rDNA. The nucleolus is the site for the transcription, post-transcriptional processing and assembly of ribosomal RNA and for assembly of ribosomes. Therefore, they are also called nucleolar organizer regions or NOR in short. Next, let us take up other nuclear bodies. In addition to the nucleolus, the nucleus contains many other nuclear bodies including Casal bodies, Gemini of Casal bodies, polymorphic interface karyosomal association or PIKA and promyelocytic leukemia or PML bodies, paraspeckles and splicing speckles. These nuclear bodies organize the nucleoplasm into functional subdomains. Let us now see Casal bodies. Casal bodies or coiled bodies are compact structures with about 0.2 micrometer to 2 micrometer in diameter, and a nucleus contains about 1 to 10 such Casal bodies. They are thought to be involved in a number of activities inside the nucleus, including RNA processing, specifically small nucleolar RNA and small nuclear RNA, maturation and histone mRNA modification. Next are the Gemini of Casal bodies or GEMs. GEMs are similar to Casal bodies in size and shape and look like twins, and hence they are named Gemini of Casal bodies. However, unlike the Casal bodies, GEMs lack small nuclear ribonucleoproteins but contains a protein called survival of motor neuron or SMN, which play a role in snRNP biogenesis. Next is the PICA and PTF domains. PICA domains or polymorphic interface karyosomal associations are nuclear bodies with about 5 micrometer in diameter and found commonly associated with dense localization of the transcription factor PTF, which promotes transcription of small nuclear RNA. Next are the PML bodies. Promyelocytic leukemia bodies or PML bodies, also known as nuclear domain 10 or ND10, Kramer bodies and PML oncogenic domains are spherical nuclear bodies which are about 0.1 to 1 micrometer in diameter and are often found associated with Casal bodies and cleavage bodies in the nucleoplasm. PML bodies contain promyelocytic leukemia protein and they form part of the nuclear matrix, which is believed to play a role in regulating many nuclear functions such as DNA replication, transcription or epigenetic silencing. Next are the splicing speckles. Splicing speckles, also known as nuclear speckles or nuclear specks, splicing factor compartments or SF compartments, interchromatin granule clusters or IGCs or B snurposomes are nuclear bodies with about 20 to 25 nanometer in diameter and are commonly found in the interchromatin regions of the nucleoplasm of mammalian cells. Speckles are generally rich in pre-messenger RNA splicing factors but are dynamic structures and their location and composition vary depending upon mRNA transcription and regulation via phosphorylation of specific proteins. Next are the paraspeckles. Paraspeckles are irregularly shaped compartments with about 0.5 to 1 micrometer in diameter present in the interchromatin space of the nucleus in some cell types, for example, human primary cells, transformed cell lines, and tissue sections. Paraspeckles are dynamic structures and are dependent on RNA polymerase II transcription. They are present during interphase and all stages of mitotic cell cycle except during telophase, where the protein components form a perinucleolar cap as there is no RNA polymerase II transcription in telophase. The next are the perichromatin fibrils. Perichromatin fibrils are usually located near transcriptionally active chromatin and are believed to be the sites for active processing of pre-messenger RNA. Next are the clastosomes. Clastosomes are small, thick, ring-shaped nuclear bodies with about 0.2 to 0.5 micrometer in diameter, formed in nucleus under high proteolytic conditions and under osmotic stress. Clastosomes are believed to be the sites for degrading proteins as they contain catalytic and regulatory subunits of the proteasome and its substrates. Let us now see the functions of nucleus. Some of the major functions of nucleus will be discussed under the following heads. The first one is cell compartmentalization. The major function of the nucleus is to form a compartment where the nuclear envelope separates the site for nuclear processes from the cytoplasmic processes. The compartmentalization helps in separating certain transcription factor proteins that regulate gene expression from physical contact with the DNA. It also prevents translation of unspliced mRNA as splicing occurs inside the nucleus and translation is carried out by ribosomes in the cytoplasm. Thus, the nucleus prevents translation of newly transcribed or unprocessed messenger RNA by ribosomes, which may result in the formation of malformed and non-functional proteins in eukaryotes. The next function is in gene expression. The nucleus is the site of transcription or the first step of gene expression in which RNA is produced from DNA template. In addition to the DNA molecule, the nucleus also contains several other proteins that play a role in regulating transcription, such as helicases, RNA polymerases, topoisomerases, transcription factors, etc. The next function is in processing of pre-messenger RNA. The post-transcriptional modification of newly synthesized messenger RNA or pre-messenger RNA transcripts takes place in the nucleus. It involves three main modifications, namely 5' capping, 3' polyadenylation, and RNA splicing. The pre-messenger RNA in the nucleus is commonly found to be present as protein complexes known as heterogeneous ribonucleoprotein particles or HNRNPs. Let us now see the conclusion. The nucleus is an important organelle of eukaryotic cells enclosed by nuclear envelope and contains the DNA molecules with associated proteins and other structures in the nucleoplasm. The internal organization of the nucleus provides the compartments or sites for many important functions of the cells. Thank you.